Welcome to the last AJP audio of 2024. I'm Aaron Van Dorn. Today on the podcast, I spoke with Ms. Devon Gray, a graduate research assistant and doctoral candidate at the University of Alabama at Birmingham, and Dr. David Knight, a professor of psychology at the University of Alabama, Birmingham. Ms. Gray and Dr. Knight have a paper in the latest issue of the American Journal of Psychiatry looking at the impact of discrimination exposure on neural reactivity to stress and psychological distress. Afterwards, AJP Editor-in-Chief Dr. Ned Kalin will once again join us to put the rest of the December issue of AJP into context. Ms. Gray and Dr. Knight, exposure to discrimination can create long-term persistent stressors that can increase the risk for psychological stress, such as depression, anxiety, and post-traumatic stress disorder. And exposure to discrimination in adolescents has been linked to increased rates of those disorders. Your study looked at stress and its link to discrimination exposure in adolescents compared to neural reactivity in young adults. What did you find?
So, we found that adolescents exposed to discrimination showed greater psychological distress, including depression, anxiety, and post traumatic stress symptoms, as well as altered reactivity distress within brain regions that support emotional function, such as the dorsolateral prefrontal cortex, inferior parietal lobule, and the hippocampus. And more specifically, those who experienced greater discrimination during adolescence showed greater stress elicited activity within the dorsolateral prefrontal cortex and inferior parietal lobule, and lower stress elicited activity within the hippocampus. We also assessed whether the reactivity to stress varies with the trajectory of discrimination exposure across adolescence and found that these same brain regions varied with the initial level of discrimination exposure, such that greater initial exposure to discrimination was linked to greater stress elicited dorsolateral prefrontal cortex and inferior parietal activity, as well as lower stress elicited hippocampal activity. Similarly, those who saw a greater increase in discrimination exposure across adolescence also showed greater dorsolateral and dorsomedial prefrontal cortex reactivity to stress. And then finally, a greater acceleration or increase of discrimination exposure as adolescents approached young adulthood was linked to greater stress elicited hippocampal activity. Now, overall, these findings suggest that adolescent discrimination exposure may alter the neural response to future stressors, especially within regions that support emotional function, which may in turn modify their susceptibility and or resilience to psychological distress.
Your study comes out of a longitudinal cohort of children and adolescents along with an imaging component. What were the demographics of the sample that your study looked at?
Participants in our study were recruited from a community sample that was originally part of a larger project studying adolescent health called the Healthy Passages Study. And initially, nearly 1,600 participants were recruited from the fifth grade classrooms of local public schools within the Birmingham, Alabama metropolitan area. Now, discrimination exposure, along with several other socio-environmental factors, were assessed when participants were 11, 13, 16, and 19 years old. We then invited 350 of those participants to return when they were 20 years of age to complete the neuroimaging portion of the study. Half of our participants were male, half were female, and about a third of our participants were white, and about 2/3 were Black adolescents, all of them from the Birmingham area.
Your study found that adolescent exposure to discrimination had long-term impacts on a number of neuroreactivity measures to stressors in young adulthood. While we don't know the exact neurobiological mechanisms through which discrimination impacts mental health, what did your findings suggest?
Well, in one of the analyses we completed, we assessed whether the stress related activity within the dorsolateral prefrontal cortex, inferior parietal lobule, and hippocampus mediate the relationships that we observe between discrimination exposure and psychological distress,
Mhm.
specifically symptoms of depression, anxiety, and post traumatic stress. These mediation analyses allowed us to determine whether including brain activity and these statistical models helps us better explain the relationship between discrimination and psychological distress. We found that by including dorsolateral prefrontal cortex and inferior parietal activity, it helped us explain that relationship. We also found that discrimination exposure was linked to greater stress related activity within these brain regions. And in turn, that greater activity was linked to lower levels of depression and post traumatic stress symptoms. Interestingly, the fact that greater brain activity was associated with lower depression and post-traumatic stress symptoms suggests that the activity we observe may offer some resilience to psychological distress.
One thing that I really liked about how we approached this research was that we assessed discrimination in multiple ways,
Hmm.
and especially like looking across the research, a lot of the times they assess discrimination as sort of a cumulative measure or sort of a dichotomous measure or maybe a trajectory, but they don't do everything at the same time. And so the fact that we have essentially three different ways that we're looking at discrimination exposure, I think was really interesting and important for this study. And it, kind of it shows how important it is to not only assess whether individuals have experienced discrimination at all, in addition to their cumulative exposure across time, but also the different patterns of their exposure across adolescence or across their lifetime.
You mentioned earlier that your study looked at around 100 white individuals and 200 black individuals. Did you consider racial discrimination as a factor in your analysis?
For this study, we didn't separate it into sort of racial groups. We are currently working on a separate project which looks at sort of more adverse childhood experiences,
Hmm.
things like neighborhood disadvantage, violence exposure, poverty. So, not quite discrimination exposure. And with that, we are doing more of a direct racial comparison. And what we do find is that in support of sort of this socioeconomic environment that we are very much aware of is that Black Americans are disproportionately exposed to adverse, childhood experiences in contrast to white Americans. And so we do see that relationship with those. I assume we'd see a very similar relationship with discrimination exposure as well. Interestingly, with that study, we found an interesting relationship with the brain, which kind of suggests this sort of resilience hypothesis where Black Americans often report lower levels of depression, anxiety, and PTSD. And they call this sort of this black white mental health paradox. And so, one of the reasons that, or what they believe is behind that, is that they are just better or have the resources to deal with discrimination exposure in a way that white Americans or those who are not disproportionately exposed to discrimination do. And so they actually showed greater activation in brain regions that are associated with lower, sort of more emotion regulation, whereas white Americans showed activation in brain regions associated with like less emotion regulation. But that is very much still, we are still running those analyses and still writing up those results. But we are looking at sort of the racial groupings in a different study, just not this one specifically.
So, following up a little bit on what Devon was saying, some of this can be difficult to assess between racial groupings. So, we assessed several different types of discrimination: race related discrimination, sex and gender related discrimination, weight, the way people talk, LGBTQ status. Okay. But if you just take racial discrimination, for example, you find many more Black Americans that report racial discrimination than white Americans.
Hmm.
That makes it tough to compare the two, right? Though we could assess things like discrimination based on weight or the way they talk or some of the other types of discrimination that we assessed. But I would also add to what Devon was saying earlier. I think that is also one of the strengths of the study is we're not focusing in on one individual type of discrimination, but we're looking at discrimination broadly across many different categories of discrimination. And then related to that, one of the things that we find is if we look more specifically at each of these types of discrimination, it seems like we find the strongest relationships with brain activity related to race related discrimination and weight based discrimination.
Your paper suggests that as a chronic stressor, exposure to discrimination might cause prolonged activation of the stress response systems and disrupt stress reactivity, perhaps influencing the onset and severity of mental health symptoms. It's a sad fact that we live in a world where discrimination is a reality, and addressing that is obviously beyond the scope of any one paper. But are there clinical implications for your findings?
So, our results support the fact that individuals who have been discriminated against are at higher risk for developing depression, anxiety, and post traumatic stress, especially when it occurs during adolescence.
Hmm.
And therefore, this may suggest a better need for early monitoring systems or early intervention systems for targets of discrimination to help protect and reduce the negative psychological impact of discrimination exposure. Also, this study has identified several brain regions that are affected by discrimination exposure and seemingly explain, or at least partially explain, the relationship between discrimination and mental health. And so, thinking clinically, these brain regions may be future targets for cognitive, behavioral, and brain based intervention strategies, such as something like transcranial magnetic stimulation.
What were the limitations of your study?
An important limitation of our work is really related to only being able to collect neuroimaging data at one time point, which was at the end of the project. Because of the nature of earlier parts of the project, details about discrimination exposure were assessed multiple times longitudinally, which allows us to assess whether and how exposure to discrimination changed over time. Unfortunately, we weren't able to assess neuroimaging data in the same way. And that limits our ability to unequivocally claim discrimination exposure alters brain function. Although we believe that is what likely will be found, future longitudinal neuroimaging research is going to be needed to determine if that's truly the case.
Is it difficult to do longitudinal neuroimaging studies of this nature due to the cost, or is it just an issue of the developing technology, or all of the above?
So it's really all of the above. When these projects were initially started, really the questions that were being asked weren't about the brain. And
Hmm.
so there wasn't somebody involved in the project to collect that neuroimaging data. But it is very, very expensive. For one participant, we're talking probably thousands, $1,000 to $2,000
Hmm.
worth of cost. When you don't include neuroimaging, that goes way down. And so
Sure.
it makes it much more feasible to collect large cohorts of participants in these studies when you don't include neuroimaging. When you do, it really expands the cost of that. There's also an additional burden on the participants when you add in neuroimaging, and that's a limit as well.
Did you find that there were a fall off in the number of people that you reached out to for neuroimaging just because of the difficulty of coming in and being able to participate in that part of the study?
Yeah, certainly there are participants who aren't going to return for neuroimaging for a lot of reasons. When we do functional magnetic resonance imaging, we're using a high powered magnet.
Hmm.
And so people who have metal in their bodies, that's not compatible with a magnet or it might not be safe for them. Those are people who get excluded right away. Even things that are safe in a magnet. So people will have braces, for example. And that's safe to put into a magnet, but it causes an artifact that prevents us from getting good quality data. So those are all people that we exclude. Certainly the kinds of things that you're mentioning are, the hassle related to it is problematic as well. Some people just don't want to spend the kind of time it takes to collect neuroimaging. We have to worry about whether people are claustrophobic. Sometimes even people who don't think that they're claustrophobic will get into the scanner and go, "Well, wait a minute, I don't like being in this tight space pulling out."
It's got to be difficult when you're doing these kinds of studies that involve hundreds of people. Each person is their own individual person, and so they might have their own individual reaction to the idea of, hey, would you get in this giant magnet tube and let us scan your brain?
Yeah, that's right.
So, what's next for your research?
This paper primarily focused on stress related brain function that is important for emotional function. However, we also believe that discrimination may lead to structural changes in the brain.
Hmm.
Specifically, we believe that discrimination may impact both gray matter and white matter. And thankfully, we have collected that data as well. And so the next steps in our research is studying the impact adolescent discrimination has on this gray matter and white matter of the brain, specifically focusing on structures involved in emotional function and their potential relationships to mental health, just like we did in this study.
Ms. Gray and Dr. Knight, thank you for taking the time to speak with us today.
Thank you.
Thank you so much.
Up next, Dr. Ned Kalin. Dr. Kalin, welcome back to the last AJP audio of 2024.
Thank you, Aaron. It's a pleasure to be here.
Earlier in this episode, I spoke with Ms. Gray and Dr. Knight about their paper looking at the impact of discrimination on neural reactivity. What can you tell us about the paper?
This is a really interesting and potentially important paper, Aaron. It really begins to get at understanding more about the biological implications of discrimination as they affect the brain and can have play out behaviorally and emotionally. What this paper does in this study is basically identify individuals that have had experiences with discrimination and looked to see how those individuals differed from other individuals that have not had experiences with discrimination. And basically, this was a study that involved about 300 individuals. About 200 of them were Black and 100 of them were white individuals. They were repeatedly studied from adolescence, from 11 to 19 years of age, from the standpoint of exposure to discrimination, whether that occurred or not. And then also a little bit later on were looked at with imaging tests to use MRI basically to assess how their brains reacted to a standardized stress test that involved providing feedback that was not positive, actually negative, when these individuals were performing math problems. And so the idea here is to look at whether or not the brain has different reactivity in these individuals that had experienced and reported experiences with discrimination. Not unexpectedly, more Black individuals reported experiences of discrimination than white individuals. But regardless of whether one was Black or white, if one did have an experience with discrimination, they had significantly greater levels of depressive anxiety and PTSD symptoms. So this is demonstrating, what we already know about the link between discrimination being a stressor and the impacts of that are related to having psychiatric symptoms, especially in the stress-related area like depression, anxiety, and PTSD. But as importantly from this study, we learned that these individuals that had experienced with discrimination also had increased brain reactivity when exposed to a standardized stressor. And this occurred in the front of the brain, in the dorsolateral prefrontal cortex, and also in some of the regions further back in the brain. So this is a early study looking at the impacts of discrimination on brain function, but demonstrating that not only is our behavior and our feelings affected, but we now begin to understand some of the substrates that may be related to that in the brain. In this particular case, the dorsolateral prefrontal cortex comes out as one of the key areas.
Sippel and colleagues have an overview in this issue of the journal looking at novel pharmacologic and somatic treatments for post-traumatic stress disorder.
Yeah, this is a really good overview, which I would encourage our audience to read. It gets at a comprehensive overview of current treatments and also novel strategies for the treatment of PTSD. PTSD has been particularly difficult to treat from the standpoint of some of the pharmacology that we use, and we've been pretty much focused on the different symptom types. And this makes that point, but also goes further in reviewing the landscape and also what we can think about maybe coming on the future, including strategies like neuromodulation.
Another review, this one from Fox and Shackman, discussing the amygdala and mental illness.
Yeah, so this whole issue is related to more or less stress-related disorders, autism to some extent, and also some of the neural substrates that have to do with experiencing stress and the impacts of stress. And the amygdala is a key brain structure that is sort of at the center or at the heart of our stress response. It lies in the medial temporal lobe. And while we talk about it as one structure, this particular review is very helpful because it gets into more detail about the complexity of the amygdala from the standpoint of the different nuclei, the different functions of those nuclei, and the different actual cell types in those nuclei. The reason that's important is because we need to now go beyond just talking about the amygdala as one thing. It would be like in the very, very, very old days, just talking about the heart as an organ and not talking about all the different components of the heart and the different cell types and so on and so forth. So this gives, I think, our readers a nice in-depth look at the amygdala in a way that is understandable, and I believe will be helpful for them not only to understand how the amygdala relates to the adaptive experience of emotion, but also how it can be involved with psychopathology that's related to stress, such as depression and anxiety, and even autism, which is not a stress-related disorder, but is a neurodevelopmental disorder.
Next, Kliemann and colleagues have a paper again discussing the amygdala, this time looking at resting state functional connectivity and autism.
All right. Well, we have two subsequent research papers in this issue on the amygdala. This is an important paper. It actually comes up with a negative finding. And we, I think at AJP, want to publish positive findings, but also negative findings when the studies are really well done and the sample sizes are adequate. And the reason for that is because that gives us a much more balanced view, I think, of what's going on in the field.
Mm-hmm.
There have been smaller studies that have come up with inconsistent findings related to whether or not function of the amygdala is altered in autism. And I want to qualify this particular study before we go any further because this looks at just one type of function. And this is what we call resting state functional connectivity, where we look at synchronous patterns of brain activity or activation using MRI across different brain regions. And in this particular case, the researchers are looking at whether or not the amygdala is synchronously in tune with other brain regions or whether there's some disconnections in relation to the way the amygdala may be connected at rest. And again, as I said, there have been findings that have been suggesting that there may be alterations in this measure in individuals with autism and other findings that have suggested maybe they're not alterations. This study is more definitive. It's a larger sample. It looks at about 212 individuals with autism spectrum disorder and also has age and sex match controls. One of the important things in this study is that these individuals were not particularly intellectually impaired.
Mm-hmm.
They may have been somewhat impaired, but not in a major kind of way. And the bottom line is that these researchers found that there was no significant difference in the way the amygdala was functionally connected to other brain regions at rest using this method when compared to normal controls, suggesting that the earlier studies that may have found disconnectivity of the amygdala at rest may not be reliable findings. Again, I'm using the word maybe, not because when we have a negative finding, it's sometimes hard to interpret that. But by and large, this is, I think, an important contribution to the literature that is leading us in the direction of thinking that this is not a measure that's going to be reliable in relation to understanding some of the pathophysiology of autism. Now, having said that, I want to emphasize that that does not mean that there are not other alterations in amygdala function that may be associated with autism spectrum disorder. This is just one measure of amygdala function.
Kamboj and colleagues have a translational paper, again looking at the amygdala, this time looking at subnuclei in rhesus monkeys.
Now, this is a much more complicated paper. And the reason we included this paper in the journal is because of our emphasis on trying to link more basic science and behavioral neuroscience with clinical science and as a best way to understand what's really going on with our patients ultimately. We need to translate back and forth between animal models, more basic research, and clinical research to really develop the best picture of mechanisms that underlie the illnesses. And this paper gets at another issue that was brought up in the review paper on the amygdala. That is to say, what is going on in individual cells in the amygdala in different nuclei in relation to how genes are working. And most importantly, what this paper does is it compares rhesus monkeys, which can be a very good model to study human psychopathology because of their relatedness to humans from an evolutionary standpoint and from a standpoint of brain structure and function. Comparing amygdala cells in the rhesus monkey to amygdala cells in the human brain to see whether or not at this level the rhesus monkey would be a valid model to understand more about the dysfunction and various types of psychiatric illnesses. I'll just say a little bit about the microstructure of the amygdala. I don't want to get into too much depth, but I do think it's important to understand some of these important structures. Basically, you can think of the amygdala as divided into two big different regions. One is the top of the amygdala, which is called the dorsal amygdala. This contains a major nucleus called the central nucleus. And the central nucleus is what we think may be a culprit in psychiatric illnesses from the standpoint of dysfunction. A particularly interesting culprit, I would say. And it's connected to the brainstem and the hypothalamus. And that's why when we get stressed and the amygdala gets activated, why we sweat and why our heart rate goes up and why we get into all kinds of postures to protect ourselves.
Mm-hmm.
The bottom of the amygdala is called the ventral amygdala, and this is consisted of regions called the basolateral regions. These are the regions that are connected to the cortex, like the prefrontal cortex, where there's information transfer about what we see in our environment, what we perceive to be stressful, and that information gets then transmitted to the amygdala, And that's where that information comes in. And then that is relayed to the central nucleus of the dorsal amygdala, where the output is, where we have these stress-related responses. So it's a little bit complicated, but it's important to understand. Regardless of that, what these researchers did is they actually took postmortem tissue from brains from humans that have died, and also from rhesus monkey brains, and looked within the individual cells in these different regions to understand whether the same genes are there and whether the same genes are working the same way. Again, as a way to validate the rhesus monkey model and to understand more about how this may provide insights into psychiatric illnesses.
Mm-hmm.
The long and the short of this study is that they found that there are many, many similarities between gene expression in these different nuclei and cells in these nuclei in rhesus monkeys and in humans, but also there were some human-specific genes that were found. And what they then did is they tried to understand which of these cell types that they identified were most enriched for genes that have to do with different psychiatric illnesses. And what they found is that there were some specific cell types in the human amygdala, especially in the lateral nucleus, that seem to be related to autism. And so this is now giving us some ideas about some of the cell types within a region of the amygdala that may be related to autism. Now, remember, I just told you before that the amygdala was not related to autism, but I also qualified that by saying that was just with this one measure called resting functional connectivity, that there may be other ways that the amygdala may be malfunctioning in relation to autism. They also found genes that were common to both the human and the rhesus monkey amygdala that were associated with anxiety disorders and depression. And these were in cells called intercalated cells. And this gets even more complicated. But these are other cells within the amygdala that have a lot to do with inhibitory input to different regions. So it's a bit complicated, but it's important to take a deep dive at times into how our brain is structured, because that ultimately is how we're going to understand the pathophysiology of the molecular alterations that are associated with our illnesses. And also, we think this will help us come up with new targeted treatments that are molecularly targeted at very specific brain regions. But importantly, in this study really validates the rhesus monkey as a species that can be used to understand molecular alterations that we know that may also be important in human psychopathology.
And finally, we have a priority data letter from Pathak and colleagues looking at post-traumatic stress disorder, women's health, and malaria medications.
So this is an interesting study. And basically what this does is this uses GWAS data from individuals that have PTSD and also have other related medical problems. And for years, we've known that PTSD is associated with many, many physical problems.
Mm-hmm.
It gets back to knowing that trauma itself can impact our physical health, and also that there may be some medical illnesses that also may increase the risk actually to be more responsive to trauma. Anyway, it's not particularly news that our psychiatric illnesses share some genetic alterations with medical illnesses. We've been talking about this for a while, and that's why we see comorbidities between psychiatric and medical illnesses. And in this particular case, what the researchers found was that there are genes that are common that are involved with both PTSD and a whole variety of medical illnesses. And also, these researchers used some statistical methods that allowed for, at least statistically, to look at what direction the commonalities may go in relation to the comorbidities. That is to say, do the genetic alterations associated with PTSD tend to predispose to and link to the genetic alterations later development of medical problems, or is it vice versa? And it turns out it works both ways. What they found was that PTSD was genetically correlated with such medical issues, interestingly enough, of having your ovaries taken out, hormone replacement therapy, having cardiovascular problems and cardiac implants and vascular grafts, having hyperlipidemia and hypercholesteremia, and even taking anti-malarial prescriptions, okay? And so this is interesting. Not sure what to make of all these specific links with things like anti-malarial prescriptions. But sure enough, what these researchers have shown is that there are genetic underpinnings that seem to be associated with the comorbidities that occur with having PTSD and having these other problems.
Well, Dr. Kalin, thank you once again for joining us.
Oh, it's my pleasure, Aaron. Take care.
That's all for this episode and this year of AJP Audio. In the meantime, be sure to check out the rest of the APA's podcasts, including the Medical Mind podcast, which is digging into a series on mental health pathfinders. I hope you'll join us anew in January 2025 for another year of the best research psychiatry has to offer. The views and opinions expressed in this podcast are those of the individual speakers only and do not necessarily represent the views of the American Psychiatric Association. The content of this podcast is provided for general information purposes only and does not offer medical or any other type of professional advice. If you're having a medical emergency, please contact your local emergency response number.